group is removed, and from here H group is removed, and this O will make a bond with the third carbon oxygen. So this phosphoric acid attached with another nucleotide will make a bond with the third carbon of the different nucleotides. So this one is a nucleotide guanosine monophosphate, and this is a nucleotide adenosine monophosphate. So the fifth carbon of the guanosine monophosphate will bind to the third carbon of the adenosine monophosphate, and here H2O is removed. H2O is removed, and this will make a bond between 3' and 5', and this bond is called in phosphodiester bond. Phosphodiester bond. Diester bond. So you can imagine the structure. Similarly, again, we can study the binding of another nucleotide. So it will make a polynucleotide. Suppose here is cytidine monophosphate is coming to bind, and the fifth carbon is CH2O, double bond O, OH, and this is O minus. Again, here is OH. So again, they will make a bond. Similarly, this is 5', this is 3'. So similarly, when we study the bonding in the DNA, each and every nucleotides, each and every single monomeric nucleotides are coming to bind together with the fifth carbon phosphoric acid group and the third carbon, another nucleotide in the third carbon of the another nucleotides. So they will make 5', 3', 5', 3' bonding in the DNA, and this is called polynucleotide chain. So this was how the formation of a phosphodiester bond between more than one nucleotide in the DNA structure or in the RNA structure. Similarly, if I'm talking about the deoxyribose sugar, this is about the DNA, and if we are talking about ribose sugar, it is about the RNA. So it was very important when we are studying the structure of nucleic acid, how the phosphodiester bonds are formed. Now it is clear how the phosphodiester bond. As I am giving you the example of formation of phosphodiester bond from 5' to 3', that means the 5' end is open for the binding of phosphoric acid or another nucleotides. Again, the 3' group is lower down, where it is again open for the binding of another nucleotides. So from upside to downside, 5', 3'. Similarly, if we are talking about the another DNA group, it will 3' to 5'. That is, the DNA is always anti-parallel strands. Two strands of DNA, both are anti-parallel from 5' to 3', from 3' to 5'. That's why they are bidirectional and anti-parallel DNA. So this was about the first bond in nucleic acid, that is phosphodiester bond. Now about hydrogen bonding. Come to the slide here, that this is a structure of, I'm making a structure of DNA. Suppose I'm making the two strand of DNA. This is adenine, make adenosine. Actually, this is not A and T only binds. Just take a glimpse. T double bond A double bond T, C triple bond G. Likewise. So both sides there is a binding of a sugar molecule. If you can observe, here is sugar, pentose sugar, deoxyribose sugar, that is pentose sugar. Both sides a sugar molecule is attached. Clear? As I told you that by the formation of nucleotide, that is nitrogenous base plus sugar plus phosphate. So there is a binding of phosphate between the two nucleotides. This is called phosphate. This is called phosphorus group binding. Similarly, likewise, this is phosphorus. Again, the binding of phosphorus make a phosphodiester bond. This is original. Actually, this is a phosphodiester bond. Phosphodiester bond, as I discussed you how it's formed. Now we are talking about the hydrogen bonding between the two nucleotides. This is a nucleotide, adenosine monophosphate, or we are talking about adenosine diphosphate, adenosine triphosphate, adenosine triphosphate. Suppose this is a nucleotide. Similarly, this is again a nucleotide, thymidine phosphate. Between the adenine and thymine, double bond is present, and in guanosine, guanine, and cytosine, a triple bond, hydrogen bond is present always. So, a hydrogen bond present inside the DNA is between adenine and thymine, adenosine and thymidine, you can say. A and T, there is a double bond, and G and C, there is a triple hydrogen bond. Come to the slide now, how it's the bond working. Because the double ring purines are bigger than the single ring pyrimidine, and the structure can only form with the purine bases, opposite pyrimidine bases. The hydrogen bond, as I told you that adenine and thymine, A and T, there is a double bond. The two strands of DNA are held together by weak hydrogen bond between the nitrogenous bases. And these nitrogen bases are like A and T and G and C. So there is a weak hydrogen bond between double bond present between them. According to the aspect of the Watson and Crick model, these hydrogen bonds are base specific. Example, adenine can only form hydrogen bond with thymine by 2 hydrogen bond. Similarly, cytosine can only form bond with guanine by 3 hydrogen bonds. Here you can study the structure, exact structure of hydrogen bonding inside the DNA. That is, a thymine bond with the adenine, a double hydrogen bond, and cytosine make a triple bond, hydrogen bond with the guanine. So this was the another types of bond, different second type of bond present inside the DNA. The major was phosphodiester bond, and another was hydrogen bond. Phosphodiester bond, it is written here, a bond between a 2 sugar group and a phosphate group. Such bonds form the phosphate sugar backbone of DNA or RNA. That is phosphodiester bond. As I discussed already, that how they are making a 3' 5' or 5' 3' phosphodiester bond, as I have discussed already on the paper. The third type of bond present inside the DNA or RNA is glycosidic bond. In DNA, refers to the nitrogen carbon linkage between the 9' nitrogen of purine and 1' carbon of the sugar syrup. How glycosidic bond? Just observe. This was the first carbon where the nucleotide, nitrogenous base, adenine, guanine, cytosine, thymine is coming to bind. So this bond, a bond formed between sugar and the nitrogenous bond, this is the bond formed between the sugar and N2 base, nitrogenous base. This bond is called glycosidic bond. And this was, here they are making a phosphodiester bond, and here the end is open to make a hydrogen bond. So finally we can conclude about hydrogen bond. We can conclude about nucleotides or DNA. There are 3 different places where 3 different bonds are formed. At first carbon, the bond formed is called glycosidic bond, where nitrogenous base is bind with the sugar. At the third carbon, there is bond making of phosphodiester bond, where another nucleotide is coming to bind with the primitive one. And here is the third bond opened with the nitrogenous base, where another nucleotide with the nitrogenous base is coming to bind and make a weak hydrogen bond. So the whole study of nucleotide, there are 3 different types, or whole study of nucleic acid biomolecules, there are 3 different types of bond playing a major role: phosphodiester, hydrogen, and glycosidic bond. Come to the PPT now. Here you can observe the beta glycosidic linkage at ninth position or 9th group of purine and the first carbon of the pentose sugar. You can observe in the figure that is denoted by red sign, that is beta glycosidic linkage present inside the DNA. Finally, a thought that education of circumstances is superior to that of tuition classes. It was told by English poet William Wordsworth. Thank you very much.
SANDHAN. All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.